thank you for the introducing me, and also for giving me an opportunity to speak here. Yeah. So actually, I am. My name is YV Nanchariah. So actually, we work on biofouling control in different cooling water systems of nuclear power plants, and we have been working on that. And then for the last 15 years, we have also added our focus on use of biofilms or aggregates for wastewater treatment. So, as we all know, microbial growth, now we have a bacteria or microbes have three types of lifestyle. So, one is the planktonic cultures, which have been studied exceptionally well. And then we have biofilms. And the third lifestyle that has also attracted interest in environmental, particularly aquatic environments, as well as engineered systems. So that is aggregates. So we have planktonic cells, biofilms, and microbial aggregates. And recently, so it has found that even bacterial aggregates play an important role in human health. So aggregates are very well known in the case of wastewater treatment, that is biological treatment. So, for example, wastewater treatment using activated sludge has been originated sometime in 1900. So it's almost a century now. That activated sludge is actually providing sanitation to the human being all over the world. So it started beginning made from UK, and then there were like much bigger aggregates, so which are like bullets, we can say millimeter sized particles of bacterial origin that has been found under anaerobic conditions, but that is not suitable for sewage, because sewage will have nitrogen and phosphorus as important contaminants. So there is like a improvement of activated sludge to granules, but that is like aerobic, under aerobic conditions. So that's what is known as aerobic granular sludge. So that's a quite recent in the sense, 2 decades ago, it was reported from Dutch, TU Delft, and TUM Germany. So there was a collaboration, and then under that, the first report was made on formation of granules from activated sludge under aerobic conditions. So this has actually like attracted a lot of interest from both microbiologists as well as the operational engineers as well as the civil engineers, because it has a tremendous potential in transforming the biological treatment or wastewater treatment scenario. So what are actually these granules are nothing but they are simply dominated by the bacteria. So in other words, bacteria plus their extracellular polymeric matrix, EPS. So if we just look into these beads, so which are actually millimeter sized particles in size, and if you look into them with the confocal or the SEM, so confocal shows that there are several independent aggregates within the granule, one particular granule. So it's like and there is a lot of matrix in that. So these granules were actually dominant or dominated by the bacteria. So these are the very clean granules because they are developed in the lab. So what you can see here. So this process, that is conversion of activated sludge to granules, was reported in 1997, and then this is in the last decade or so, it has been translated into a technology which is known as an radar technology from TU Delft, and then a company has funded the research. So if you see this two glass tumblers here, one of the tumbler is filled with activated sludge while the other one is filled with the granules. So this shows how fast or so granules settle much faster, as well as it occupies less space. So it shows the compactness of the biomass. So this essentially helped in actually improving the treatment, not only treatment, as along with that reducing the wastewater treatment infrastructure as well as the cost. So as a result, now it is considered as a treatment choice for full-scale systems in different parts of the world. So, but So all good when it comes in the lab actually. So lab they can be generated very well under defined conditions, under defined carbon sources, etc. But when it went to the field, especially when we go for a real wastewater treatment like sewage, municipal wastewater, which is complex, not only complex, but it is also not so strong. So under those conditions, it was found that this formation of granules is slower, as well as it takes very long time, and granulation, extent of granulation was less. So there were also concerns, how stable are these granules, so how long, so if there is a breakup, and then that can lead to loss of biomass, failure of the system, as well as the deterioration of the treated water, so which is not desirable. So then I'm bringing another one. So when it comes to, so far whatever I have mentioned is concerned with the sewage, which is from the domestic wastewater or the tap potable wastewater. So salinity is not a problem. But when we are talking about saline wastewaters, so saline wastewaters or saline sewage or industrial effluents, the common approach is to either adopt activated sludge or the granules to high salinity conditions gradually. Thereby, we can increase the salt tolerant bacteria, and then finally we'll be able to have a hallow tolerant granules, which does the job. But there are several challenges here, because it is very difficult to achieve ammonia removal in saline conditions, because nitrite often accumulates in the system, and there are hardly much work on P removal, especially BioP, what I mean. So there were also reports that when the granules are exposed to salinity, they lose their stability, they undergo breakup. So there were also, as I mentioned, long startup or adaptation periods are required. So, so now uh, we are. I'm coming to the alternative to whatever I mentioned, that granulation from activated sludge is a norm. Okay, we always seed the reactors with a lot of seed sludge in the beginning. That is activated sludge. So here alternative is, so this whatever activated sludge formed to granules is the normal route. So whatever alternative is, let's say wastewater or water has a microbes already. So why don't we start from that source instead of taking activated sludge and then adapting it to higher salinities? So and then try to granulate them and see whether it does granulate and perform the required treatment. So that is what I mean is the granulation from scratch. So that is what is the de novo granulation. So what we tried is, so we have taken saline source, that is seawater as a source for microbes in our case. So we made synthetic wastewater from seawater. I mean synthetic wastewater here is, we just spike the seawater with required nutrients, that is carbon source, ammonium, and phosphorus. So then we investigated granulation as well as the biological nutrient removal. So we try to address granulation of the bacteria which are present in the seawater, and then removal of organic carbon, that is acetate in our case, and then nitrogen, ammonium removal, as well as phosphorus. Under saline conditions, like 34 ppt was taken, because the seawater salinity is 34 ppt. And then later we also investigated how these granules which were formed at 34 ppt will respond to the higher salinities and then perform nutrient removals as high as 120 ppt. So approach is very simple, as I mentioned here. So we have a seawater has a different groups of microbes, and when we tested, it has about 10 power 3 to 10 power 4 CFU per ml, and then so that water was fed into the container, and then this container was operated as a sequencing batch reactor, because SBR is the best one for achieving granulation or aerobic granular reactor study. So we operated the reactor with 6 hours. So 60% of the treated water is removed and fed with the fresh medium. Again, it is a seawater based medium. So if I'm showing here the actually growth during the first 1 week. So since we did not add any inoculum or seed in the beginning, so water, whatever the feed, so synthetic wastewater, so it became turbid during the first week due to the growth of the microbes present in the seawater based medium. And then that growth actually slowly it has increased in terms of size of the particles or patches. Initially they were like patches, and then so that is what is shown in the images here. So first 1 week, but once what it has happened is there was a growth of the microbes, and then these microbes tried to aggregate and then formed patches. These patches actually they were retained in the reactor. So then they were subjected to shear force as well as the further growth, which led to the bigger formation of the bigger particles in the reactor. So that is what is shown here. So this is what actually we found in the seawater. Is the type of cells that were found in the seawater medium, which are actually giving into the reactor. So if you see the actually temporal development of the particles here, so within week actually we have seen a luxurious growth of the cells, and then that led to formation of patches, and which actually became much denser later, and then formation of a bigger particles, which are like millimeter sized particles. So which was actually not expected. It was like a surprise for us, because within 10 days we have seen a lot of dense formation of dense particles in the reactor without actually using any seed sludge. Then further characterization of these aggregates shows that these were actually comprised of cells along with their matrix in them. And of course, we also saw some precipitates in these particles. So because when we are working with the seawater, as we know, it all contains a lot of salts. So that is so the calcium containing precipitate formation was observed. So then coming to the performance was monitored in terms of TOC removal, nitrogen and phosphorus. So as shown here, the influent values were shown here at the black line. At the below is almost like from beginning we are seeing actually TOC removal. And coming to the ammonium, we are giving about 30 ppm of ammonium into the system, and then so within 10 days a good removal of ammonium was established in the system. So this was really interesting to us. So it's not just ammonium removal, and then accumulation of nitrite and nitrate were very minimal. So that showed that it is a good system for complete N removal. And then phosphorus removal was a little slower in the beginning, and then it took some time, but it has improved significantly. But if we compare this with the freshwater systems, this is still far better than the freshwater systems, because freshwater systems still take much more time. So as compared to CN, P removal usually takes time, establishment of PPBR. What I mean is BioP removal by enhanced biological phosphorus removal pathway. So what essentially we have seen is, so I have showed earlier that the granulation was rapid, and that was coincided with the effective or establishment of BNR pathway, that is biological nutrient removal pathway, both carbon as well as CNP removal. And then if you see the single cycle, so whatever I have shown here results for almost 80 days. You see the time scale is up to 80 days, and each data point is from a 6-hour cycle. And then here within the 6 hours, how the removal is taking place. So as the 6-hour cycle is usually operated with, first hour is the filling, and then remaining time we have aeration followed by the settling and a decant, which is common to SBR. That's how the SBR is operated. So giving an anaerobic phase is very crucial for granular reactors. So that's how we actually always give the initial anaerobic phase or static filling, we call it. So during this filling and then so we see the COD removal pattern, or TOC, say total organic carbon removal pattern, and then ammonium removal is coincided by the formation of small concentrations of nitrate as a minute concentrations of nitrite. So we don't see accumulation of nitrite. Usually the saline systems, whatever when activated sludge is adopted to saline conditions, often accumulation of nitrite has been reported by the several authors. So such a case has not been found in this case, because probably that these microbes are actually already growing in that environment, saline environment. So we are probably selecting the right organisms. So then P removal, if you see, which actually typical for EPBR mechanism. So that is under anaerobic conditions, it actually releases the PAOs, phosphate accumulating organisms, polyphosphate accumulating organisms. What they does is, as part of their metabolism, they release small amount of phosphorus during the anaerobic phase, and then during aerobic phase, subsequent aerobic phase, they accumulate much more phosphorus than what they have actually released. So that's how we see a net P removal from the system. So this is actually typical. It shows that the P is removed by a EPBR mechanism under saline conditions. So whatever data I'm showing here is this 34 ppt. That is what our synthetic seawater based medium. So, and then coming to the bacterial community, we analyze the granules for different physicochemical characteristics, etc., which I'm not going to discuss here. But what actually we found that there is some amount of reduction in the bacterial diversity as the granulation progresses. So that means, as compared to the seawater, what actually we are feeding into the reactor, and then what was actually formed in the reactor with time. We compare granules versus seawater microbiome. We found much lesser diversity in the granules. So that's how what is expected, because there is like a selection and enrichment of organisms in the SBR configuration. But nevertheless, what we found is there was like a fair amount of diversity in the system, and also several organisms which can do NNP removal. So that's why they are able to remove NNP removal from the synthetic wastewater. And then, so this is whatever I have presented until now is at 34 ppm. Then we wanted to see how these granules will respond to higher salinity. So then we went to gradually we increased the salinity to 120 pp up to in the reactor. So these are the granules harvested or collected at different salinities. We see some morphological differences in the granules in the SEM images, suggesting some kind of precipitation also. This is because of hyper salinities that were used. However, the granules were stable at all the salinities, as well as they were showing good settling characteristics. And then we also found that the nitrogen removal, let us see here, nitrogen removal, and at different salinities ranging from 34 to 120 ppt. So the below one actually shows that effluent levels are much below than what is desired. So this is what is important here to note, that irrespective of the salinity, higher salinities, N removal was not affected. Ammonium removal was not affected. And then if you see single 6-hour cycle, so the typical cycle shows that ammonium removal black line, and then these below lines are nitrite and nitrate. Very small amount was observed, and this blue line shows percentage removal with respect to time, 6 hours cycle. So this shows actually that though granules were formed at 34 ppt, they were able to tolerate, not just tolerate, and they were able to perform well at hypersalinity. Interesting thing is to note that no nitrite accumulation in the system. And then P removal was also similar pattern was observed. Though there were little disturbances at higher salinities, but it was actually stabilized at, as it was adopted to higher salinities. So similarly, the EPBR removal pattern was maintained with removal during the release during aeration phase, as well as uptake during the aerobic phase was found at all the salinities, indicating that P removal, P was mainly removed by biologically and through EPBR. So we wanted to actually check whether how much of it is available. Question can come. It is under hypersalinity when the experiment was done. So for that, actually, the fed batch experiments were done. So first one is TOC removal, and then next plot is nitrogen removal, as well as P removal. Each bar represents one experiment, actually, fed batch 24-hour, and next batch like this. So almost 5 batches we repeated to actually check reproducibility. So TOC was so first TOC, N, and P removal. First 3 graphs were actually conducted with the normal granules. So whereas the fourth graph data shows the granules with the heat killed granules. So here normal granules, there was no problem as they were already removing. However, with heat killed granules, percentage removal was very marginal. So only P removal little bit was observed. So all otherwise, so this shows that actually the removal was mainly driven by the biological process. And then coming to the part of the granulation, try to actually based on the microscopic observations. And then, as I mentioned, so we are starting from the cells here. So not the activated sludge. So cells also present in the water that is fed into the batch reactor. So these cells, when they are fed within 6 hours, they are trying to grow, and then with subsequent cycles, this growth was retained in the system, and then that retained growth was growing and then becoming into the bigger and bigger particles. So that is the growth, and then attachment and aggregate formation, and then growth of these aggregates and their retention and further growth. So of course, detachment is part of the overall process, but this is how the granules normally are developed in the sequencing batch reactors. So other steps are like, so these first steps are different from the conventional granulation process, because in the case of conventional system, granulation is started always from a bigger aggregates, all above 100 micron activated sludge blocks. And interestingly, in this system, we have not seen deterioration in the settling properties or formation of flocks. So because the system was configured in such a way that growth of or retention of aggregates and granules was favored. And then coming to the removal mechanisms, so based on the chemical data and microbiological data, so ammonium, as I mentioned, ammonium was removed, as well as no nitrite and nitrate accumulation. And then during ammonium removal, the formation of or the release of ammonium nitrite as well as nitrate to the bulk liquid was minimal. So that's how we actually proposed it to be like a partial nitrification. That is like ammonia is getting converted to nitrite, and then which is getting denitrified directly. Although we see some amount of nitrate formation, and that also gets denitrified. So, and then P removal is by EPBR. So that is, so in the anaerobic phase, P is released, and then in the subsequent aerobic phase, P is uptaken or accumulated in the cells. So, and apart from EPBR and our XRD, the SEM, EDAX, as well as the P fractionation measurements showed that some amount of phosphorus is present as precipitates. So there is some amount of phosphate precipitation, chemical precipitation by other salts which are present in the seawater, like calcium and other minerals. So what actually, so finally, I would like to conclude here that so what actually has shown is so previously, so it is like the activated sludge or seed sludge used to be the granulation from seed sludge used to be the norm. So here we are exploiting or exploring granulation from the planktonic microbes which are present in the water. So we have taken seawater as the model system because we wanted to investigate nutrient removal under halophilic conditions or salt conditions, saline conditions. However, the same concept, let us say, is for a sewage. Sewage is rich in the desired microbes which are required for wastewater treatment. So it is so we have also so instead of activated sludge, one can directly go with a sewage granulation from the sewage microbes. In fact, that we have done already, and we are one of the full-scale plant is working in Kalpakkan based on the similar concept, where actually the full-scale system was directly fed with the wastewater without using any seed sludge in the beginning. So then within a week's time, we were able to see growth as well as the formation of aggregates and then granules in the system. So advantage is that so what happens is that sometimes sewage treatment plants like our place in India, majority of the plants of activated sludge process are conventional systems which uses two tanks. One is aeration tank, second is settling tank, and this system is not so good for removing or efficient energy removal. So in fact, they are not performing some of these plants. So when I go for activated sludge from such system which does not do BNR, NNP removal, and then I will have difficulty in establishing, it takes time. So in such a system, when I go for, let us say, right from wastewater, which is rich with NNP organisms, then establishment of NNP removal was found to be much faster in the system. So coming to the concluding part, so what we have observed is it is possible to make the granules from the planktonic microbes, mixed culture microbes, and then this has been applied to synthetic seawater here, and it can be very well applied to other wastewaters. And then there are other benefits here. And then coming to, moreover, one important point I would like to mention is this. This is like a more simplified, can be a simplified model for understanding granules, because activated sludge becomes more complex for understanding the process. Here, since we are starting with the cells, so in that sense, it will be like a model for us to understand the granulation part. And then, so I would like to thank here my colleague, Mr. Sarvajit, who has done the experiments as part of his PhD in our lab. And then, of course, I also would like to thank all our office staff and researchers, etc. And then those who contributed to this work. And then atomic energy, because I work in Department of Atomic Energy, Bhabha Atomic Research Center, for their generous funding and facilities as well as the resources.
Thank you so much. So, thank you, Dr. Nanchariah. I think we will conclude so that we have some time for questions. William, you can read out the questions.
Right. So the first one we've had come in, someone wants to know what the retention time of the SBR was. So this was back at the beginning of your presentation.
Yeah. In this case, we are giving 8 hours retention.
If anyone wants to come in, which wastewater category do you think is best suited for this type of treatment?
Actually, this work was done with the synthetic, that is seawater, spiked with the nutrients. So, but it is suitable for treating saline effluents, saline wastewaters. And then we have also applied same concept to municipal wastewater. And that actually, we now have a full-scale plant which is treating about sewage from 320 households operating plant. So there also we have not taken activated sludge. So we started from sewage microbes, whole process. Yeah.
Great, thank you. That's the two that we've had come in. If anyone else has any questions for any of the speakers, please do put them in the Q&A box. We've got one more there. We've got time to do one more. The last question, do you think you could apply to treat marine fish production sludge?
Could you repeat?
Do you think that your system could be applied to treat marine fish production sludge?
Yeah, because this is this can treat the saline 34 or even higher salinities, as I mentioned. In fact, we have also decreased the salinity up to 17 ppt because we wanted to check the saline sewage treatment of saline sewage as well.